Good afternoon to everyone. I welcome all of you to the 79th lecture in the lecture series in nonlinear dynamics conducted by the Department of Nonlinear Dynamics, Bharathidasan University, with the support from RUSA 2.0. It is my pleasure to introduce today's speaker, Dr. Shakti Menon from Institute of Mathematical Sciences, Chennai. Dr. Shakti Menon obtained his PhD from the University of Sydney, Australia in the year 2008. He then moved on to Queensland University of Technology, Brisbane, Australia, and carried out his higher studies. He came back to India in the year 2012 and continued his academic career at the Institute of Mathematical Sciences, Chennai, and right now he is serving as a project scientific officer there. Dr. Menon is working on different topics in complex systems, including studying emergent phenomena in complex systems, analyzing pattern formation in development biology, and investigating collective motion in living systems. He has substantially contributed to the development of this field. He has presented a large number of talks and published several notable articles in his area of expertise. With this short introduction, now I invite Dr. Menon to deliver his lecture. Over to you, Dr. Menon.
Thank you so much for the kind introduction, and also thank you so much for inviting me to give this talk. It's really my pleasure. Just let me, I today would like to talk about one of the projects that I have been working on at IMSC for a couple of years now. So this is some work that I have been working on with a PhD student, Chandrasekhar Koyamuri. He has recently finished his PhD and now he's pursuing a postdoc at Harvard. And of course, this is also in conjunction with Professor Shitabra Sinha, who I've been collaborating with for many years at IMSC. I have organized the talk in the following way. I'll briefly give an overview of the general area of developmental pattern formation. I'll talk a little bit about the specific type of problem that we are interested in, which is contact-mediated cellular communication, and explain about how this particular mechanism plays a really crucial role in developmental pattern formation. And then I will cover two of the problems that we have been working on. One is trying to develop a unified mechanism for describing somitogenesis, or the formation of segments in vertebrates, and also how tissue growth can be, in some sense, regulated through the disorder in the cellular arrangements, which is a very important problem in deciding when organisms need to stop growing. So I will explain this. And if there's a time at the end, I will just very briefly cover some of the other problems that Chandrasekhar, myself, and Professor Shitabra have been working on. So to start with, the embryo, from its very early stages, it undergoes a lot of different steps as it continues to grow. So it's basically starts off as a bunch of cells, and over time you have processes such as cell division. So the cells will divide, and that's essential for the organism to grow. Cells can migrate to different parts of the embryo, perhaps in response to certain chemical signals. So this is what's known as chemotaxis. You could have programmed cell death, which is known as apoptosis. So that's also important in certain contexts. And perhaps most importantly, one of the most important processes that is involved in this stage is differentiation. So cells can differentiate to take on specialized roles, and as the embryo develops into eventually into like an adult organism, these form the different types of cells which constitute the different sorts of systems that are within the body. So this is also a very key step. An essential part of this process is self-fate determination. So essentially, the cells, they need to be able to decide what to do. Now, like depending on where the cell lies in the embryo, some cells may need to perform a certain task, whereas some cells in another part may need to perform a different task. So, for example, in the late-stage embryo, which I've shown on the side here, you'll see that you'll see the beginning stages of the somates, of which eventually will give rise to the vertebral column. You see that long sort of tail, and you have the beginning stages of the somates forming. So within the somate, as I will explain in one of the examples later, some cells need to form like the front end of the somate. Some cells need to become the back end of the somate. So how do they know which they're going to which fate that they need to attain? So this is a very important problem, because the cells by themselves are not they don't have any sort of coordination center that's telling them that you perform this task, you perform that task, right? So the cells somehow need to use the information in a very smart way to be able to decide what they need to do. And essentially, the big problem in development is how cells actually can be able to decide their fates depending on the kind of information that they receive. You could say. So one of the models, or conceptual models, I should say, that has been proposed to describe the sort of developmental pattern formation is this famous French flag model, which was proposed by Wolpert. And so this was one way by which cells can use information from their surroundings, and based on that information, decide on their eventual fate. So in this schematic that's shown on the side, so you have a xenopus embryo, and you can see that there are there's a sort of a concentration gradient shown from one side to the other of some chemical. This is just like the concept. So there is some gradient, and then you see that cells towards one side are blue, cells towards another side are red, and in the middle that they are white. So how does and so towards one side they'll form maybe like they have a dorsal fate, a ventral fate, So so this is a kind of a crucial sort of step. But how do they make this decision? Because the cells by themselves don't know, am I in the middle, am I in the side, am I in the left? Because they are just cells. How do they know? Right? So the way that it's been proposed that they obtain the so-called positional information is by identifying the concentration of morphogen in their vicinity. So, for example, the blue cells will see a very high concentration of morphogen, and they know that it's above a certain threshold, and they'll say that, okay, if it's above a certain threshold, then I need to perform a certain fate. So in this case, they have to perform the so-called blue fate, whatever that means. The cells in the middle will notice that the morphogen concentration is below the threshold, but above another threshold. And then they could say that I need to perform fate number 2, which could, in this case, is colored by white. And then the cells on the right, they find that the morphogen is below both the two thresholds, and so they perform the third fate, the red fate. So essentially, this is like a smart way by which cells can utilize information from the surroundings and be able to figure out where they are. And knowing where they are, they can then decide what to do. But of course, this is only an idealized sort of scenario, because in reality, the morphogen gradients are not so smooth, you could say. Like if you have the production of some chemical towards one side, and it's diffusing across the embryo, you would find that if you measure the actual gradient of this morphogen, it would be quite noisy. Right? So, for example, near this boundary between the white and the blue, some cells would see a higher morphogen, some cells would see a lower morphogen. So you'd have actually a very fuzzy kind of boundary. In fact, you could get a very distorted sort of shape. You would not look like a kind of a neat flag like what you're seeing over here. And this can actually have very important functional consequences, because in a developing embryo, the precision is paramount. If you have imprecision in these boundaries, then you can have pathological outcomes. You could have a deformed sort of shape, and this is obviously not wanted. So the cells need to have some mechanism by which they can surmount these potential problems that can arise. So they need to be able to have other ways by which they can get information that allow them to still make good decisions, even though the environment might be noisy. So how do they go about doing these sorts of things? One way by which they do that is through intercellular communication. And in the case of developing embryos, there are two main ways by which they can communicate. One is the so-called paracrine signaling, wherein they produce signaling molecules, and these molecules can diffuse from the cell and move to other cells in its vicinity, and it can induce changes in those cells. The second way is through juxtacrine signaling, wherein these when cells come in close contact with their neighbors, they will have a binding between the ligands and receptors on their surface. Now, I'll get into the precise details of one example a bit later, but basically, ligand receptor coupling is it can be understood as follows. So you have ligands on the surface of one cell, which will bind to receptors on the surface of another cell. And once this happens, when you have a successful binding, then this will trigger some activity further downstream. And so in other words, the activity of one cell can affect the activity of a neighboring cell. In the case of development, there are two main ways by which this happens. So in other words, there are two main sort of signaling mechanisms which involve the sort of ligand receptor coupling. These are the so-called notch and hippo pathways. And I will, coincidentally, in the two examples that problem sets that I will talk about, I will cover notch and I will cover an example of notch and of hippo signaling. So first of all, let's take a look at notch signaling because this is perhaps a little bit more complicated. So the notch signaling pathway, essentially what you have is, as I said, you have receptors on the surface of the cells. And these receptors have essentially two sort of domains. So one is sort of protruding outwards from the cell, and another one is sort of lying inside the cell. So the one that's lying inside is called the notch intercellular domain, NICD. Right? And when the notch binds successfully with the delta of another cell, then the NICD will split away from the notch, and it will translocate to the nucleus, where it can either upregulate or downregulate the expression of some target genes within the cell. So this is you can think of it something like a kind of a lock and key mechanism. So effectively, when a notch finds a delta of another cell, then it will have a successful binding, and it will cause the NICD to perform its task. So this is what's called as transactivation. The other, you can also have notch when it binds to the delta of its own cell, then nothing happens. So it's able to know that it should not. There should be no sort of further activity when it binds with the delta of its own cell. So that's called cis inhibition. But let's not get into that for the time being. The main thing is the transactivation, which is an important mechanism that I will talk about in the next problem. So having sort of covered all the basics, let's take a look at the first problem. So this is a video showing the development of a zebrafish embryo. So as you can see, you have a growth happening along the tail. And as the tail is beginning to grow, you can start to see these sort of like segments that begin to form. I'll play the video again so you can see it clearly. So take a look at the sort of segments and that are beginning to form, and you can see that over time you can get more and more segments that are beginning to form closer towards the tail. Now, the formation of these segments, as you can see, is happening in a very regular and timed manner, right? So if the segments form too quickly or they take too long, then this can cause a problem, because then you can have segments which potentially may be too large or too small, and this can lead to again a pathological outcome. So this is not something that you want. So this is again a very precisely timed process that needs to be well regulated. And so basically during this process of somitogenesis, you have the so-called the PSM, which is the part of the mesoderm, the pre-Semitic mesoderm, which has not yet formed somates. You can see that in the sort of in the picture above, the blue region, that's the PSM. So over time it gets smaller and smaller because more and more somates are beginning to be formed over there. Of course, the tail itself will also grow. So this is it's not so it's a slightly more complicated process. So this is as I said, a very well timed sort of process. And what is the sort of process that could potentially sort of act as a kind of a clock for this? And so it turns out that there are these sort of genes whose expression level occurs in a very well-timed manner, very much in line with what you would expect to see. So I'll play this movie again. So you can see that in this video, you can see that the expression level of certain genes are sort of changing in a kind of oscillatory sort of manner across the PSM. And while it's doing that, you can see gradually a few somates are beginning to form. Unfortunately, it's a very short movie, so you cannot see it too clearly, but you get the idea. And so it's believed that these clock genes are those which underlie the sort of precise nature, the well-timed nature of this phenomenon. And so people have tried to come up with a kind of a conceptual sort of framework to explain how exactly this could be happening. Like, what are the processes that are involved? One model it's not, shouldn't really call it, well, it is a model, but it's not a mathematical model. It's more like a conceptual model. For explaining this is called the clock and wavefront model. This was proposed a long time back. It's, as I said, it doesn't specify any molecular or cellular details. Basically, what it's saying is that you have these sort of autonomous cellular oscillators, as I showed before in that video, and their activity is arrested when it encounters some wavefront. This is an abstract wavefront that will freeze the oscillators that it encounters, and this will leave a record of their activity at the time of the arrest. And so the frequency of oscillations is now going to be important, right? Because if you have a wavefront that just moves from one end to the other, and remember, this is just a wavefront in the sense that it freezes the activity. So let's say we freeze the activity from one side to the other, and you have fast oscillations versus you have slow oscillations, then essentially you find that the segment length is going to be different for the two, because essentially the wavelength, you could say, is different. And so this becomes so in other words, so this is a way by which people have tried to explain this, but this is as I said, it's a conceptual framework, and you could even go so far to say it's like an outline for a framework, because it doesn't actually specify how exactly this happens. So it's saying that these are the necessary criteria for a framework that will explain everything. You can see, I mean, in practice, this is something like how it would look like. So here is a schematic for showing an embryo, and you see that you have the growth of a tail, and then you have the wave of clock genes expression towards one end, as you move towards away from the tail towards the anterior side, there you start to see that the somates are forming. Now, when you have a low frequency, or in other words, a longer wavelength, you see that the spacing between the somites in panel B below is quite large. On the other hand, if you have a very fast, high frequency oscillations of clock genes, there you can have somites that form like more somites that are very kind of closely spaced. And this could and it is proposed that this could explain differences between different types of vertebrates, because the gene expression level is different. So the frequency of gene expression level is different in different vertebrates. But again, so far we don't actually know. We don't actually have a quantitative explanation for what exactly is happening here. We know that this is that this is that these factors are related, but how does it all work together? So it was proposed a while back that maybe the way we should be thinking about this is in terms of a model with 3 tiers, right? And so in the bottom tier, you have basically these oscillators whose activity to determine the period of the clock. So this is what we just looked at in the video. Then you have mechanisms through which the different cells synchronize to form these sort of waves. And finally, you have the mechanisms that arrest the oscillations and form the somite-somite boundaries. So it was proposed that if you look at it from this perspective, then you can actually have theoretical and experimental investigations separately for each of these 3 tiers, and you can try to see if this your proposed model is valid. The way we were looking at it was that, well, is it possible to have just one model that can in some sense capture the mechanisms of all the 3 tiers? And that's what we were trying to attempt in this work. So in the schematic you see here, so this is similar to the in some sense to the schematic that was shown a couple of slides back. With the difference is that now we are labeling 4 different regions, and we are showing hypothetical sort of time series for gene expression levels of 2 adjacent cells. So the light blue and the pink correspond to the expression levels of a pair of cells that are neighbors. Now in region 4, where it's synchronized, you can see that the activity of the blue and the green are the same. In the region 3, you have traveling waves. In region 2, the activity is being arrested, so it's frozen. And in region 1, you see that it is frozen, but they are at distinct levels, which corresponds to distinct cell fates. Right? And so these could potentially mean something like the cell which will form the rostral part of the somite, the cell which will form the caudal part, which means just the front and back part of the somites. Right? So what we were hoping to do is we can have one model that can in some sense dynamically capture the sequence of transitions going from 4 to 3 to 2 to 1. So we have one mechanism that can explain this sequence, because essentially that's what you see in practice. In practice, the posterior, the tail end is what is growing, and then you have this sequence of dynamical states, and then finally you have the somites. That's stage the number 1. That's what we have marked. So that's the final stage. So we want to go from 4 to 3 to 2 to 1. So how do we go about this? Well, what we have is a very simple minimal model that can try to describe some of the behaviors that you would expect to observe. So to begin with, we just start off with the minimal model of a genetic oscillator. Right? And so we just say that we have so of course we wanted to oscillate, so we need to have at least two genes. We have X and Y, and we say that X is an activator, serves as an activator, right? And Y serves as an inhibitor. And this circuit is capable of oscillating. And then we also have to have the interaction which is occurring through this notch signaling. Now remember from the schematic that I showed earlier, when you have a successful binding, what happens is that this the intercellular part of the notch will move to the nucleus. Let me just show you again actually. So when you have a successful binding, the intercellular domain will move to the nucleus, and then it will target the, so it will activate the target genes. And in this case, we know what the target genes are, X and Y. So we have some, so here we have some proxy variable, we call it NB. And so this is created when you have a successful binding of notch with the DTR. TR is trans binding, so the D of a neighboring cell. So when notch binds to D of a neighboring cell, then you have this NB, which will upregulate the activity of X and Y. Okay? And finally, to complete the circuit, we also need to have the X and Y inhibiting the activity of the suppressing the production of delta ligands. And this is something which I won't go into now, but this is something which is biologically known. And so for this reason, we have taken this particular motive. So this essentially will complete the whole circuit. So we have with these 5 variables, we can describe the activity of so I should say we can describe the interaction between neighboring cells in this tissue. And so we actually I should mention one more thing, that this model is of course a very simple model, but it was described in the following paper, which is mentioned at the bottom. And notch delta signaling, we have adapted a framework that is described in the paper mentioned at the bottom. So we have, in some sense, we have merged the 2 models together, and doing so, we end up with this model. And so I've colored the various parameters of the model in 3 different ways. So we see that you have the parameters shown in green. So these are the parameters of the uncoupled genetic oscillator. And then, so in other words, if you have no coupling, so these are the only parameters that matter. They will control the oscillations. And so these parameters we just take from the original model. We have not changed them all that much. So it turns out that these parameters are not so important for our final results. And similarly, the parameters in blue, we have more or less taken from the original paper, because they turns out that they do not affect the result all that much. These are the parameters of the notch and delta interactions. And I mean, I won't go into step by step of each term in the model, because basically I've already described the framework over here. It's essentially, we have just transcribed this. I mean, the model has just transcribed these interactions into equation form. But the key thing to note here is that we have the 3 parameters in red, the F, G, and the H. And these are the parameters that mediate the coupling. So the effect of notch binding, of notch delta interactions between a pair of cells is governed by these 3 parameters. And so these in a subsequent slide, I will show what happens as you change these 3 parameters and the different types of patterns that you can obtain. And just before I leave the slide, I just want to mention that, so we here in the model, we have DTR and NTR. All that means is that we have taken the average value of notch and delta of the neighboring cells, which is as a consequence of transbinding. Because for any cell I, it will have some neighbors J, which can go from 1 to NGH. And when, of course, I mean, as it turns out, for all the simulation that we show, we either have a pair of cells or we have a chain of cells. But so which means it will be either 1 or 2. But in any case, but the point is, the DTR is just the average value of DTR and NTR, the average values of D and N of the neighbors. That's it. All right. So moving on, when you have a pair of cells that are coupled through this mechanism, you can actually get a wide range of dynamical patterns. As it turns out, only some of the patterns are important for the actual somitogenesis process that we're describing. But nevertheless, it's important to be able to distinguish between them when choosing the right parameters of F, G, and H, the coupling parameters. So we need to be able to distinguish between them. And so for this purpose, we follow like a kind of a flow chart that will sort of help us to distinguish between the different types of patterns. I won't go into the details of the flow chart, but essentially all it's showing is that we first of all have to be able to say whether you have oscillations, and if there's no oscillations, then is it a homogeneous steady state or an inhomogeneous steady state? And then if they are oscillating, are they oscillating is it exact synchronization, or is it a different type of synchronization? And if it's not exact synchronization, then what type of synchronization is it? Right? So as I said, the details are perhaps not so important for now, but this is just to say that we need to be able to distinguish between the different types of patterns. And once we are able to do this, then we can scan the parameter space for our coupling parameters F, G, and H, and we can see what are the kinds of patterns that we obtain. So what these figures show is essentially because these are parameters which we don't know what values they should have. So we something which we need to explore. So when we explore these parameters, what we observe is that you can get, as you can see, a wide range of dynamical patterns. But the thing is, it turns out that for the process of somitogenesis, we want one very specific sequence. We want a sequence that follows this, going from 4, 3 to 1. Right? And so, can we actually have one follow a sequence like this? Now, it turns out that there is such a path. So if you fix your parameter F and your parameter H, and then you just vary the parameter G, then it turns out that you can actually, and that's shown in the bottom right figure. You can go from this dark blue to light blue to green and so on. So that effectively corresponds to exact synchronization, homogeneous steady state, inhomogeneous steady state, and so on. This might seem a little bit arbitrary why we're doing this now, but this will become very apparent later why we have chosen this. And secondly, you may ask the question, well, why did you just fix F and H and vary G? Why could you not vary the others? That is also possible. We could also fixed G and F and varied H. And it is possible that we could have come up with a similar sequence. But for now, this is the sequence that we have chosen. And also, now that we know that we can fix F and H and vary G, you can see that in the model we have labeled GI, which means that G is now changing from cell to cell. This is actually important because what this is saying is that each cell is going to experience a slightly different value of G. And the way that we have motivated this is that different cells are subject to different concentrations of morphogen. Remember, all of this is happening in the presence of morphogen gradients. And I will show an experimental result later that explains how important this is. And so this, as it turns out, is actually very crucial for us to be able to get this result, because if G does not change over space, then you're not going to get this expected dynamical sequence. So having said all that, and now that we understand how this works, let us see what happens when G is different. So we are still not yet at the stage where we are reproducing the full dynamical sequence. We still want to first understand what is the effect of changing G. So to do this, we have a pair of coupled cells with the two values of G. And what we do is we change the two values, the difference G1 minus G2 of the two cells. And of course, we can also change the mean value of G, right? Changing G1 and G2 will in turn change the mean G, and it will also change the difference. I'm sorry, I should have said it a little bit more clearly. Yes, so as you change your G1 and G2, one can actually see that interestingly, the difference G1 minus G2 doesn't matter as much as the mean value of G. So you can see in the top figure, when the mean value of G is large, so maybe above 3 to 6, then you can see that you only get one pattern, which is the steady state pattern, regardless of your G1 minus G2. On the other hand, when G1 minus G2 is very low, you only get one pattern, which is exact synchronization. But for intermediate values, potentially you can get a third pattern, inhomogeneous in-phase synchronization. But that's not so important for now. the point is that overall, the mean value of G is what is the key here. And similarly, what we find, actually, sorry, I think my thing is blocking. Let me just move this. Yeah. The mean value of G, as you can see that when you increase the mean value of G, you observe that the difference y1 minus y2 of the expression levels y. So remember, y is the inhibitory gene. So the difference y1 minus y2 becomes larger. Now, why am I mentioning this now? Again, this is something that becomes very important in a subsequent slide. So this is the effect of the changing the parameter G. So now, how do we put it all together? So, like I said, the G, our motivation for varying G is the fact that this is blocking this again. Yeah, the motivation for changing G is the fact that you have gradients of these morphogens. So, what is a morphogen? Comes from morpho, which is form or shape. So morphogen means that they are responsible for the creation or the generation of shape. So these morphogens are known to play a very important role during development. And in the case of the zebrafish embryo, you have many morphogen gradients which belong to different families. You can have fibroblast growth factor, retinoic acid, and so on. And in particular, the retinoic acid one has a peak at the anterior end, and it decreases as you move towards the posterior end. And this, it turns out, is very essential for us because the other two are their main role is to sustain oscillations, which is perhaps not so essential for our framework. We already have oscillations. So we will consider this particular morphic gradient. So we have a gradient which is higher at the anterior end, which is away from the tail, and then it decreases as you move towards the tail. Which means that you can say that the values of G1 and G2 should be higher as you move away from the tail. So that's what we have to incorporate in our model. So this is the last step, and then we'll have put it all together. So the last step is this. Remember that this tail is growing over time. So in other words, if you have some block of cells which are in the tail, then over time they'll no longer be right at the very end. They will be somewhere now in the middle. Right? And as a consequence of this, they are observing a morphogen gradient, but the slope of the gradient which they are observing will change over time because now you're having more and more cells, and so the gradient is also evolving as well. So from their perspective, the gradient looks like it's varying over time. So if you want to model what these cells are doing, then you have to have a gradient which changes over time. Right? So this is what we incorporate. So now we have, so not only do we change G, which is important, as I said for the previous slides, the G also has to change over time. And what we do is, so basically we have G, it varies exponentially over space. So space is I, so it changes exponentially over space, and it also changes over time. And so this is the way that we do it. So basically Gi equals Gmin times exponential of some slope lambda G times xi. And lambda G is, that's fixed. So the slope is, the lambda G is fixed. But the xi is what is changing because now the position of where the cell is now going to be changing over time. So incorporating all of this, we now have a dynamically varying GIFT. So what happens when we put this into a model? So let us see what happens now when you have two cells. So when you have a pair of coupled cells, this is what you observe. Initially, you'll find that you have synchronous oscillations, and then you have the cessation of activity, and then after some time, you find that slowly the two activity levels will begin to diverge. In other words, that delta will increase. So you have over the course of one simulation, you have all the different dynamical states that you would expect to observe in somitogenesis, starting off with synchronized oscillations, traveling waves, cessation of activity, and the divergent activity. And furthermore, although it's not so apparent in the figure on top, if you look at the time period and see how that changes over the course of this this interval, you'll see that actually it's a gradual increase in the time period as well. And this is, which in other words, the oscillations are slowing down. And this is something which is also in agreement with experimental observations. So all of this is good. So far it's saying that for the case of two cells, we are able to capture the set of dynamical transitions that we observe. And before I move on to what happens in the full tissue, it's just to quickly say that in reality, of course, you have noise. So we want to confirm that the results do not change when you incorporate noise. And so for that, we introduced a noise term in each of the equations, where basically the noise term is the product of that variable with some theta times the noise term. And what we find is that the results do not change much at all. I mean, in other words, we get exactly the same qualitative trend that we obtained earlier, as you can see in the figure below. And if you crank up the noise strength, I mean, eventually, so what these figures are showing is that we have shown a very large number of trials, and the solid line is the average of those trials. So you can see that, I mean, of course, if you really crank up the noise, then you can see that the results start to get quite messy. But at least for the case of small noise, the results are pretty good in the sense that they still show the same qualitative trend that you see for the case of the deterministic case. So that's good. And now finally we see what happens when you have a tissue. So in the case of a tissue, what we see is that well, the way we modulate it is that we now have 20 cells, although the number is not important, but we have 20 cells, and they are coupled to their neighbors via notch delta signaling. And we find this block of 20 cells is now moving up a gradient. In other words, the G is time varying. What do we see? So initially we start off with exact synchronization, and then we begin to see that there are some waves for a short-lived period. There are some waves that are in the moving across the tissue. Then the waves die out, and then over time you can see that you have the formation of these segments. You can see that you have temporally invariant high low sort of states happening at the end. So in other words, you get something very much like what you would expect to see in the case of somites, where alternating sort of cells are going to a high and a low state. And the figure down below is just showing the same result in a slightly different visual representation, but it's the same thing. So this is great. So in other words, means that we have now been able to have one mechanism that can capture all of these dynamical states and up to the point of the formation of the different somites. One question which kept coming up whenever we were submitting this paper for review was, this is all good, but what about the fact that you just have a somite of length 1? Like, what if you want a somite of length 2? Because you could have a bunch of cells forming a somite, not just one. As it turns out, as you change the parameters, you can actually get slightly different type of segmentation patterns that are forming. So you have the exact same dynamical sequence initially, but then at the very end, you can get like different types of clusters that are forming. Now, this is, as I said, the parameters do not change the qualitative nature of the transitions, but they do play some role in the final pattern that we observe. Of course, the parameter space is enormous, and we have not been able to explore it that carefully. But at least this gives us some hope that maybe that it is possible to have a regime where you can get somites of larger size, and there could be other explanations as well. Like, for example, you could have multiple morphogen gradients that are at play. So, in summary, this this work, we were able to have, as I said, one mechanism that through which notch-delta coupling of cells, which are subject to a morphogen gradient in an expanding pre-symmetric mesoderm, can actually capture all the expected dynamical transitions that one would expect. And this also it in some sense it is able to capture the three different tiers of the framework that is proposed to synthesize the so-called clock and wavefront mechanism. And of course, there is more work that can be done on this, but hopefully our work can make a contribution towards understanding this a bit more. And the publication for this paper is listed below. So before I move on, and since it's already 4:15, maybe are there any questions on this topic?
Students, do you have any doubts? Students, if you want some clarifications, you can rise. Yeah, maybe you can continue, then we will come back.
Sure. Okay, so I will go on to the next part. The next part is a bit shorter than the first part, but please let me know in case I'm going over time. Okay, so I'll move on. Okay, so so the second problem is related to growth arrest. Now, one of the most important sort of capabilities that tissues need to have, especially during development, is when to stop growing. Right? So for a tissue to know how to grow, that's easy, but a tissue also needs to know when to stop growing. Right? And when it does not stop growing in time, you can get a pathological outcome. So the example that is shown over here schematically is in the case of a Drosophila imaginal disc, and the wild type, which is the normal scenario, which is shown in the corner, is what you expect it to look like. But in the larger panel, where you have the overexpression of one gene, is giving rise to a scenario where sorry, just 1 second. I think is that a question?
There is a question in the chat box.
Okay. And what basis I chose notch and hippo as examples? Okay. So the reason that I chose notch and hippo, so notch and hippo are in fact the only types of juxtacrine signaling that are important during development. So in the case, as I mentioned, in the case of development, there are two types of signaling mechanisms: paracrine signaling and juxtacrine signaling. Now, I think if I remember correctly, there are totally like 11 types of signaling pathways that are involved during development. 9 of which are paracrine, 2 of which are juxtacrine, and the 2 juxtacrine ones are notch and hippo. And since the focus of these studies are juxtacrine or contact-mediated signaling, I have chosen these 2. Okay. So moving back to this, yeah. So this can lead to problems if the tissue doesn't know when to stop growing. It turns out that this Yorkie or this YKI, this gene, which is overexpressing here, is an important transcription factor in the hippo signaling pathway, which I just now mentioned. And this has also been implicated in cancers and things like that. So it is a very important pathway. So getting back to the question, okay, so how do tissues know when to stop growing? Right? So cell division, as you probably know, it's a natural part of the cell cycle, and so this occurs during this mitosis stage. And to prevent the cell division, it requires the oscillations to be stopped. So if the cell stops, I mean, the cell cycle oscillations stop, then sometimes you can stop the cell from dividing. Right? And when you have an increase in cell density, as a consequence of that, more of the cell surface area is in contact with adjacent cells. And so you have more sort of potential binding between receptors on the 2 surfaces, and it's proposed that this can trigger like an intercellular signaling cascade, which will repress the oscillations, right? So over time, what could happen is that you will see that the oscillations will slow down, and then eventually they'll stop. And then so once it reaches, so you can see that there's a kind of a correlation between an increase in density and a reduction in cell division. So in some sense, it's sort of nicely tied together. So basically, when it's very sparse, the cells can grow and divide without any constraint. But then as it gets more dense, then they start to slow down, and then eventually they stop. Right? So this is a kind of a nice way of thinking about potentially how this could be happening. Of course, there is an issue with this, which I will get to in the second part of this discussion. But for now, this particular mechanism is something which one can try to model. So you could the way that we modeled it is we have like a motif comprising three cyclically repressing genes, and where one is self-activated. So that is the motif ABC that I've shown over here. Now, why this particular motif? The reason for that is because it turns out that this particular motif has a very useful property in that it can show a near invariant amplitude over a wide range of frequencies. Right? So it can oscillate at a range of different frequencies, but the amplitude will not change all that much. And this is actually very important for this context because it's known that the cell division rates can change a lot within the same organism. So obviously the oscillation frequency is related to the cell division rate because that tells you how fast the cell is going to divide. But you don't want the amplitude or the expression level to change all that much. So this is why we chose this particular motif. And okay, so we have that, and then you say that the cells can bind to ligands of the neighboring cells. And so when you have a successful sort of binding between cells, then this can trigger this hippo pathway. So we don't model the full details of the pathway, but we just say that as a consequence of this binding, you'll have a downstream effector molecule S. So this will be sort of produced. And S, effectively, it has information about the extracellular ligand concentration. And this, we know that it has a repressive role. We know that basically it tends to sort of slow down the oscillations and eventually terminate the oscillation. So we want S to repress one of the molecules. So we make S repress C. The results we have verified don't vary much whether you which molecule you vary. You can also you can also inhibit A or B. It doesn't matter. But for these simulations, we have chosen to inhibit C. Okay? And this is how this is just a mathematical representation of the motif that of the diagram that I showed earlier. So effectively, you have A, B, and C are going to inhibit each other, right? So you have so each equation has two parts. One part is the conversion of the inactivated form to its activated form, and the second part is a term that describes the inactivation of a species through the binding with its repressor, which basically means that C is repressing A is repressing B is repressing C. So that's it. That's all that we're trying to model. Right? So it's fairly straightforward, and we are using sort of terms with So basically, we're using these terms, and in addition, we have two extra terms. One, we have a self-activation term in the equation for A because that's what we have in this schematic over here, and in addition, the equation for C also has a term that describes its inactivation by S. Right? So we have that as well. And you have the parameters K1357. They correspond to the rates of activation transitions. K246, so they're the inactivations, and K8 is the downregulation of C by S. So this is K8 is actually the important parameter because this tells you the information what's coming from the outside. So this is, as we'll see, it displays a very important role. Now, I won't go into details about the parameters, but we did yes?
But these equations are linear?
Yes.
They're linear equations.
Yes. Yeah. So we kept it as simple as possible because we just simply wanted to describe, to see if we can have a sort of in the absence of any information from the outside, you just have a simple oscillatory behavior. And then now in the presence of this extra S, this is going to change the frequency of oscillations and eventually slow down. So yeah, that's it. So we kept it very simple. What we can do, so we have, remember, so here, if you notice, we have all these Hill functions. Now, I won't go into details of all the parameters. We have explored that in the paper. But the main point I want to mention here is that we have these Hill functions, and if they're sufficiently steep, you can approximate them as a step function, right? So that's what we said, that let us see if in the limit where it's extremely steep, what if we model them as step functions. So that's what we have done over here. So we said, okay, so now we're just going to convert all the Hill functions into Heaviside step functions. And now that we have done this, we see that, well, effectively, we have 8 different regimes that can occur depending on whether the A, B, or C are greater than or equal to this K. Right? So K is basically the term that occurs in these equations. So you can effectively write down 8 different sets of models that describe the dynamics in each of these regimes. Right? So for example, if A is less than K, B is less than K, and C is more than K, then you can effectively write down the set of equations on the lower left. But if A is more than K and the B and C are less than K, then you can write down those other equations. So this is like a reasonable approximation for what's happening in those regimes. Now remember, A, B, and C are changing over time. So that is not to be forgotten. But all we are saying is that when these conditions are satisfied, then these dynamical equations provide a reasonable approximation for what is happening in these cases. And why is this? Why did we try to make this approximation? And the reason we try to do that is because what we can write down once we have this simplified sort of setting is that we can obtain these state transition graphs. Right? So we say that so let's just say we have a binary string to represent whether A, B, or C are greater than or equal to K. Right? So then we have the different possible strings, and then we can say that, well, what is the so remember, in each of these cases, you can see what does it evolve to. So in the case A less than K, B less than K, C more than K, you can evolve the dynamical system and see, well, now what happens. And then it turns out that this can evolve to a scenario, for example, where maybe A is now more than K and B is less than K and C less than K. So you can see where it goes, and you can track this, and it turns out that depending on the value of K8, you can get 2 different outcomes. So when K8 is less than some threshold value, and I won't derive the precise form, but actually you can see how this K8 star is obtained by looking at the figure below. But like I said, I won't get into that now. But depending on whether K8 is more than some critical value, you can actually get 2 different types of state transition graphs. So in one case, you see that starting in one dynamical regime, you can travel around and you form a loop. So basically you keep oscillating, and when K is more than K8 star, you see that eventually you go to a steady state. You eventually end up in a scenario where you have A more than K, B less than K, and C less than K. And of course, this can be verified now that we understand this. Now that we understand that K plays such an important role, we can go back to the full system and see that the results actually are obeying. So so this is not simply speculation. This is actually what the full system is doing. So this is just a tool which we can use to help us to understand what the full system is doing as you change K8. Now, as I said, so maybe before I get to the next part of this, let me just mention. So let me just recap. So what have we learned so far? So what we have learned is basically the parameter K8 star. K8 is tells us the information about the strength that a cell so the strength of the it tells us information about about the extent to which the cells are in contact with neighboring cells, and that will affect the extent to which the C is inhibited, to which the oscillations are suppressed. So in reality, there is a problem, because the problem is we have been assuming so far that the density is changing over time, but it's not always the case. So in epithelial sheets, the cells are always in contact with others, or for the most part, they're in contact with other cells, right? So here is an example where you see that this is an epithelial sheet which is evolving over a couple of hours, and the density is not changing. All that's changing is that the geometry is changing. In other words, that the cell shapes are becoming more regular over time, and perhaps the packing structure is becoming more organized. That is what is changing, not the density. So in other words, the idea that you have more neighbors coming in and that's going to stop the cells from oscillating, it doesn't work. So we need a slightly different way of trying to understand this. So the idea that we had was, well, we know that the cell shapes are becoming more regular over time. So maybe we should use this information about the cell shapes in order to modify the kind of signal that the cell is receiving from the hyposignaling pathway. So that was the idea that we had. The way that we tried to model this was the following. So first of all, we need to create an irregular lattice and be able to sort of model how it becomes more regular over time. There's actually a very nice way to do this, which is known as Lloyd's algorithm. So I'll show you the movie. So you can see we start off with an irregular lattice, and then over time it's become more regular. Let me play it again. Yeah, and so after so many iterations of this procedure, you can see that the story is becoming more and more regular. The way that this, I won't get into full details about this because I don't have too much time. But very briefly, the way that the algorithm works is that we choose some n seeds that are distributed throughout the lattice, and effectively we are trying to find the the seeds are used to generate the so-called Voronoi tessellation, which is what we are viewing here. And we are trying to find the centroids of these and then move the seeds closer to the centroids over time. And then the idea is that eventually, if you keep following this procedure, you will end up in a regular lattice. Now we calculate that the centroid is obviously it's not something that you can do perfectly. So there's a procedure by which that you follow this. So I won't get into the details for now, as I said. So but nonetheless, you follow this algorithm, and we find that we can get this lattice going from irregular to a more regular structure. And now that we have this, what we do is we modify this S, the downstream affecting molecule. We say that S is now going to depend on the concentration of ligands that are on the cell surface, which is a function of its perimeter. Because now if a cell has a smaller perimeter versus having a larger perimeter, then the concentration of ligands is obviously going to change, right? So that is obviously going to have an effect on what the S is going to be, because then S is going to get a stronger or a weaker signal depending on how many ligands there are, right? So this is what effectively we have done here. We have also verified that what if this is, rather than consider the perimeter, we consider like the area, or we consider like the degree, which is the number of neighbors of a cell. And it turns out that the results qualitatively do not vary all that much. So so we'll for the time being just consider the perimeter, and we'll see what the results look like. So so as you see, so here is what we get for the Lloyd algorithm. It's similar to what I showed in the movie before. So initially you have an irregular structure, and then over time you have a structure that's more regular. The colors essentially represent the number of sides of that polygon. So if you see in the figure on the left, you have more like a variety of colors, which means that you have different types of shapes. But the figure on the right, the colors are much more uniform, which means that the shapes are much more regular now. In other words, you have cells of maybe size 5, 6, and 7. They are the most common types of cells. So that there is an increase in regularity. And and so then what we find is that successive iterations of this leads to a narrowing. As I said, it leads to a narrowing of perimeters, which implies a more homogeneous distribution of such cell shapes. If you consider the frequency of cell cycle oscillations across the domain, which is something that we can measure, this provides a nice proxy for the growth rate of the tissue. So we effectively look at, so we can say how fast the tissue is growing by knowing what is the frequency of cell cycle oscillations. So we measure that, and then we look at how this changes over different Lloyd iterations J. So basically, when J equals 0, it means it's disordered. As J becomes larger, it becomes more ordered, right? And we can track how this changes as a function of K8 as well. So in the figure below, you see that we're showing how R changes with J as well as with K, right? And interestingly, we observed the following, which is that if you look at the two dashed lines, you see one dashed line is increasing and one dashed line is decreasing, depending on which value of K8 it is. And this is interesting because, and this actually connects back to our analytical result, which tells us that the kind of result that we get is going to depend strongly on the K8 value. So looking at this a little bit further, we see that as we increase the homogeneity, which means the J becomes larger, we see that the fraction phi of cells that stop oscillating is going to increase when K8 is strong, and the reverse is true when K8 is weaker. Right? So what does that mean? So it means when K8 is, so when you have a stronger signal from the outside, more cells are going to stop oscillating. When you have a weaker signal from the outside, the reverse is true. In fact, you're not going to get much change in the cells are going to be oscillating more. Right? So that is a nice result because that connects to what we found analytically. And furthermore, we see that if you look at the frequency and the dependence of the frequency on the parameter, we see that the critical size at which the oscillation stopped changes as a function of K8. So these two values, by the way, the two values of K8 I should have mentioned are arbitrary. I mean, the precise values are not important. The only point is that one is much larger, one is much smaller. And this, we see that depending on the K8, the critical value at which the cells stop oscillating is going to change. Now, why is this important? Well, because when you change we know that when you make it more homogeneous, the distribution is going to change. It's going to become more narrow. But if you look at the critical value, what we calculated from this previous figure over here, so when K8 is 1.53, you can see the critical value is much larger. And you see that when K8 is larger, when K8 is 1.53, you see that the shaded region is going to occupy less space, less area for the red curve compared to the purple curve, right? So if you take the cutoff value for the two different curves, you see that, which means that less cells are going to stop oscillating. On the other hand, if you look at here for 2.56, if you take this critical value, you see that now at that critical value, if you look at the area under the curve for the pink curve versus the purple curve, you see that much more of it has stopped oscillating. So in other words, an increase in the homogeneity is going to cause more cells to stop oscillating. Right? So it's a complete opposite result that you're obtaining. So this is the key here. So in other words, the value of the K8 is going to not only depend. It's not only going to decide what the outcome is going to be, but also the geometry is going to play a big role in deciding whether or not cells are going to be oscillated or are going to be arrested. So so that takes so that is the so this work basically the main result is that we have found a causal relation between the increase in regularity of the arrangement of the planar arrangement, basically how disordered it is, and the growth arrest. Yeah, so that's basically it. And so then this is the publication that we have. I suppose I'm probably out of time, but so then I will just very quickly just mention that we have also tried a few other different problems. I guess I won't get into them now, but I just had a one slide sort of snapshot of each of them. So we had also looked at the case of the how notch delta signaling will affect the nature of spatial patterning in a and avoid, and how it can be used to change the size of domains in a regular manner. And we've also looked at the case of how disorder can impact the lateral inhibition, which plays an important role in determining cell fate. And we find that it can actually play a big role. And most recently we've been looking at what happens if you have, in addition, the noise. So in other words, it can notch delta signaling help to sort of attenuate the effects of noise in terms of making sharper boundaries. So I won't say any more about that. So finally, once again, I just like to acknowledge my collaborators. So as mentioned, Dr. Chandrasekhar, who this is his PhD work. So he was the main driving force, and so he deserves a lot of the credit. Professor Shilabra Sinha, who I have been working with for many years, and he's been extremely supportive. And finally, for the second project which I mentioned was in collaboration with Dr. Fernando Casares at CABD in Civil. So with that, I'd like to thank you.
Thank you, sir. Sir, there is a question in the chat box.
Yeah, sure. Okay, so I took the frequency independent amplitude model. What if we take a frequency dependent amplitude model? Will they follow the Hippo pathway? Frequency independent amplitude. No, so I think there's a slight confusion. Now, what I took was the case where, okay. Yeah. No. So So what I mentioned by this is that here if you have a motive of this nature, the frequency will you can change the frequency of oscillations without having to worry too much about changing the oscillation amplitude. Right? Now, the reason we did that is because it's actually important to change the frequency, because as I mentioned, in different types of I mean, so within the same organism, it's been reported that you can have much variation within the cell division rates. Right? So this is something that should change. Something that we don't want to change that much, however, is the amplitude, because we don't want the expression levels to now also change as the amplitude is changing. Right? So as the frequency is changing. I'm sorry. We don't want the expression level to change as the frequency is changing. So that's the reason why we chose this. So in other words, if you have the reverse, if you have the frequency being fixed but the amplitude changing, I mean, you can study that, but that's not going to be able to describe this particular process that we want to be looking at.
So any other questions from students? Students, you need some clarifications or doubts? So you are not receiving any questions. So, okay. So maybe they will write to you. I don't know.
Sure.
Okay. So since we are not receiving any more questions, I would like to conclude the session by thanking Dr. Shakti Menon for accepting our invitation and giving a wonderful lecture. Thank you, sir.
Thank you so much.
I will write to you the other details by email.